Welcome to MLSQR. Our topic is systemic lupus erythematosus for the clinical lab. Part 1 includes a brief intro, pathophysiology, immune tolerance, hypersensitivity, and SLE autoantibodies. A red malar or butterfly skin rash may appear during active episodes with flare ups with ultraviolet light triggering an episode. Genetics, hormones such as estrogen, environmental toxins, certain drug side effects, infectious agents with molecular mimicry with cross reactivity with self antigens are thought to play a role in the etiology of SLE. Mutations in certain genes of the major histocompatibility complex with certain human leukocyte antigen types are thought to lead to an imbalance of the otherwise finely tuned immune system of susceptible individuals. If one twin has lupus, then the other twin may have a 1 in 4 chance of developing the disease, suggesting that genetic and environmental factors influence the prevalence. Women are more likely to be affected with lupus than men by a ratio of 9 to 1, suggesting that estrogen may play an important role. In severe cases, the red erythema rash may lead to a wolf like appearance, hence the name lupus erythematosus came about. Usually, cells lead a normal life until they reach apoptosis, programmed cellular death. With release of nuclear antigens, including DNA, both single stranded and double stranded, histones, and other nuclear proteins. SLE is thought to be caused by a breakdown in both B and T cell tolerance that results in the production of a diverse collection of IgG antibodies, which recognize a wide range of self antigens, including DNA protein complexes, and RNA protein complexes. B cells and plasma cells are activated to make these autoantibodies. Immune complexes with anti-nuclear antibodies and nuclear antigens trigger the complement cascade. With recognition, activation, and cytolysis mediated by the membrane attack complex, leading to a hypersensitivity reaction, which is type 3, attracting more and more of the unwanted neutrophils and macrophages, leading to inflamed tissue damage. Here we see IgG antigen complex formation with complement recognition on the antibody Fc receptors, with complement protein depletion during active disease, and deposition of ANA immune complexes with autoantibodies that are anti-nuclear, leading to systemic manifestations, such as the butterfly malar rash, discoid rash, and photosensitivity. Neurologic disorders include seizures and psychosis. Serositis includes pleurisy or inflammation of the serous membrane surrounding the lungs. Pericarditis, inflammation of the serous membrane surrounding the heart. Autoimmune hepatitis, oral ulcers, esophagitis, peptic ulcers, lupus enteritis. Lupus arthritis involves joints with tenderness, swelling, and sometimes effusion with limited range of motion. Hematologic manifestations include hemolytic anemia, leukopenia, thrombocytopenia. Hypocomplementemia stems from C3 and C4 consumption by immune complexes. A serious complication of lupus arises when immune complexes deposit in the glomeruli with glomerulonephritis. Ominous signs are urinalysis with the urine protein and blood in the dipstick and hyaline and RBC casts in the urine microscopy. Let's review briefly the types of immune hypersensitivity reactions with type 1, type 2, type 3, and type 4. The mechanism of type 3 hypersensitivity involves formation and deposition of immune complexes that are not cleared by macrophages. where we see complement depletion with CH50 and C3 diminishing. This table efficiently summarizes the types of hypersensitivity reactions, their mechanism, and some examples. Many different autoantibodies cause autoimmune disease. SLE is caused by autoantibodies against single stranded and double stranded DNA, also antihistone, anti Smith, anti Rho, and anti NRNP. Lab tests that may be valuable to assess for the systemic manifestations include CBC, SAD rate, CRP, LFTs, BUN creatinine glomerular filtration rate, and urinalysis. Anti-nuclear antibody with immunofluorescence may be valuable, as well as CH50 and C3 complement levels, and this will correlate with disease status with active disease as the complement is depleted and diminished. Specific autoantibodies against nuclear DNA, RNA, ribonuclear proteins, and phospholipids are detected in patients with lupus, as listed in the column to the right. Be sure to watch our next tutorial video for part 2. We'll present test methodologies that are more specific for systemic lupus erythematosus, including ELISA, ANA immunofluorescence, lupus anticoagulant, and coagulation mixing studies. And now for the questions and answers, which are similar but not exactly like the ones in our ACP study guide.